Dear viewers, greetings. In this video, we are going to see about single beam spectrophotometer. This video contains the following contents. First, introduction about single beam spectrophotometer, followed by principle of single beam spectrophotometer, instrumentation of single beam spectrophotometer, working of single beam spectrophotometer, applications of single beam spectrophotometer, advantages of single beam spectrophotometer, and finally limitations of single beam spectrophotometer. First, single beam spectrophotometer. A single beam spectrophotometer is a type of spectrophotometer that uses a single beam of light either in the visible or UV range to measure the absorbance of transmittance of a sample which is contained in a cuvette. In a single beam spectrophotometer, the intensity of light is measured before and after it passes through the sample. The design of the single beam spectrophotometer is simpler and more affordable when compared to the double beam spectrophotometer. Principle of single beam spectrophotometer. By applying Beer-Lambert's law, which relates the absorbance of light to the concentration of the analyte, the concentration can be calculated. In a single beam spectrophotometer, the instrument measures the light intensity that passes through the blank or reference solution and the sample solution separately. First, the intensity of light is measured with a blank cuvette, usually containing just the solvent or buffer. Then, the intensity of the light is measured after it passes through the sample. The absorbance is calculated using the same formula as a single beam spectrophotometer, A equal to log I0 by I, where I0 is the intensity of the light through the reference or blank, I is the intensity of the light through the sample. Instrumentation of single beam spectrophotometer. The instrumentation of single beam spectrophotometer contains 5 major parts. It includes light source, monochromator, sample holder or cuvette, detector, and finally output display. First, light source. Typically, a tungsten lamp for visible light or a deuterium lamp for UV light provides a continuous spectrum of light. Second, monochromator. Monochromator is a device that selects a specific wavelength of light from the light source. It typically uses a prism or diffraction grating to isolate the desired wavelength. Third, sample holder or cuvette. The sample is placed in a transparent container called as cuvette where the selected wavelength of the light passes through the sample. The fourth one is detector. After the light passes through the sample, the detector measures the intensity of the transmitted light. The spectrophotometer compares this transmitted light with the intensity of the original light to calculate the absorbance. Finally, output display. The absorbance or transmittance is displayed digitally or on a chart. Working of single beam spectrophotometer. First, the light source emits a spectrum of light and then the monochromator selects a specific wavelength. The selected light passes through the sample cell and the detector measures the intensity of the transmitted light. Finally, the readout system displays the absorbance or transmittance value. Applications of single beam spectrophotometer. The first application is general laboratory analysis. Spectrophotometer or the single beam spectrophotometer is suitable for a wide range of applications including quantitative analysis, qualitative analysis, and kinetic studies. The second application is educational purposes. The single beam spectrophotometer often used in educational settings to demonstrate the principles of spectrophotometry. The third application is routine measurements. The single beam spectrophotometer is suitable for routine measurements where high precision is not required. Advantages of single beam spectrophotometer. The first advantage is simplicity. The design of the single beam spectrophotometer is straightforward, making it easy to operate and maintain. The second advantage is cost effective. Typically, the single beam spectrophotometer is less expensive when compared to more complex instruments like double beam spectrophotometers. The third advantage is good for routine measurements. The single beam spectrophotometer is suitable for routine laboratory works where high precision is not critical. Finally, limitations of single beam spectrophotometer. The first limitation is less accurate over time. Since the reference blank and sample are measured at different times, there may be drift in the light source or detector performance over time, and it affects the accuracy. The second limitation is time consuming. The operator needs to manually measure the blank and sample in succession, which takes more time when compared to a double beam system that measures both simultaneously. The third limitation is sensitive to environmental changes. Variations in the ambient conditions like temperature and light intensity between measurements can affect the results. Dear viewers, that's all about the single beam spectrophotometer. Thank you for your support. Thank you.